Okay, now that we have our carbohydrates, our dietary carbohydrates digested by our pancreatic and intestinal enterocyte enzymes, let's look at carbohydrate absorption. So let's draw ourselves a simplified version of an intestinal epithelial cell. And we'll put the lumen of the intestine at the top. So this is where our glucose, galactose, and fructose are located. Again, remember the glucose originated from starch. The glucose also originated from sucrose, which is the combination, the disaccharide fructose and glucose, and also from lactose, which is the disaccharide galactose and glucose. So now that we have these larger molecules digested, we have to get these inside the body. The way we're going to do that is by transporter activity. We need transporters because remember that these are highly polar molecules, and polar molecules will not pass across the plasma membrane because the interior of the plasma membrane is hydrophobic. So in order to get these monosaccharides across the plasma membrane, we need a transporter. The transporter we're going to use is called SGLT1. This stands for sodium, glucose, transporter, type 1. This is going to take glucose from the lumen of the intestine to the inside of the enterocyte, into the cytosol. Because it is sodium glucose transporter, it is coupled. So this is a symporter, which means it's going to take two individual solutes and move them in the same direction. Again, remember how these transporters work. The transporter only works when glucose and sodium are bound to the outside of the cell. Then they will transport them inside the cytosol. This is also secondary active transport. So this will operate by a gradient, and if we have a large dietary component inside the lumen, you can figure that the high concentration in the lumen of glucose and sodium from our diet is going to help drive the process here as these are moving down their concentration gradient. But that's not sufficient because this is our primary energy source, and so we are going to couple this to primary active transport. And once again, we will see here our familiar sodium, potassium, ATPase, which is our primary active transporter. So this here is going to hydrolyze ATP using the energy of ATP to pump sodium outside the cell. Now, of course, we have a potassium channel to allow potassium to be released from the cell so that it can be reutilized in this transport process. So the bottom line, what we're going to have here is a depletion of sodium inside the cell, which is going to increase the concentration gradient of sodium from the lumen to the inside of the cell, which is secondary active transport using the gradient established by the primary pump to establish this gradient of sodium, which is going to drive glucose in. Now, what about galactose? Well, galactose is also going to be used by the same transporter, SGLT1, because remember, the structure of Galactose is almost identical to glucose, except it has a hydroxyl group in a flipped position in the ring structure, which makes it an epimer. But the structure of galactose and glucose are so similar that the SGLT1 will recognize either sugar and transport it in this method. The last sugar we have to consider is fructose. Fructose has its own transporter. This transporter is called GLUT5 for the glucose transporter type 5. Okay, that's odd, right? Glucose transporter transporting fructose. It was a misnomer. So when these glucose transporters were identified, GLUT5 was originally thought to transport glucose. Turns out it only transports fructose. So fructose is going to be moving down its concentration gradient. So this process is called facilitated diffusion. Diffusion works by having a chemical or electrical gradient, in this case a chemical gradient, high fructose in the lumen because of dietary source, low fructose inside the cell. That gradient will drive this process. It's facilitated by a protein transporter. So GLUT5 will bring fructose into the cytoplasm. Now once we have them into the cytoplasm, some of the glucose will be metabolized by the enterocyte. Remember that the enterocyte has to generate a large amount of ATP for its normal function. For example, driving this pump in order to get nutrients inside the body. The enterocyte also has to undergo regeneration, and we have stem cells that are located in the crypts of lubricant, and these stem cells also require an enormous amount of ATP. So the epithelial cell of the intestine is going to have first dibs on grabbing our energy source, making ATP. But the bulk of the glucose that enters the cell is going to exit the cell by another transporter. This transporter is called GLUT2 in the intestinal cell. Stands for glucose transporter type 2. This operates also by facilitated diffusion. So this transporter will work again on a gradient. So the glucose is going to be very high inside the cell, resulting from the glucose sodium transporter, and the glucose is going to enter the blood and essentially be swept away, which will keep its concentration low. So the GLUT2 transporter will operate effectively because of the gradient established. What about fructose? Well, GLUT2 in the intestinal cell turns out to be a promiscuous transporter, which means that it recognizes both glucose and fructose. This is promiscuous because, remember, glucose is a 6-membered ring, fructose is a 5-membered ring, so the structures of these two are very, very different. However, this particular GLUT2 in the intestine will allow these to go through, and fructose is gradient is going to drive this facilitated diffusion and get the fructose into our blood. Now, the importance of understanding this is exactly the principle of rehydration therapy. So what is rehydration therapy? Well, this is important for individuals that have cholera. And remember, cholera is when we have a defective chloride channel, because the chloride channel is kept open because of the cholera toxin, and we have a flood of ions into the lumen, which is going to pull water into the lumen of the intestine, giving us our watery diarrhea. So in order to combat this, what we need to do is we need to ingest glucose plus sodium. So this is the principle of Gatorade. So Gatorade, fluids, has glucose in it in the form of sucrose, and it has sodium ions in it, and then by having the substrates for the SGLT1, we are going to take both of these osmotically active molecules, bring them into inside the cell, and when these are pulled into the cell by SGLT1, water will follow. And thus we'll be able to get water from the lumen inside the body for rehydration. As a quick note, this also is a very popular treatment for hangovers. Hangovers due to alcohol consumption causes dehydration, and so in order to prevent the hangover because of the dehydration, consuming glucose with sodium will increase the hydration and counter the effects.